My topic of presentation is bioaugmentation of soil artificially polluted with acetaminophen and hydroxychloroquine. Pharmaceutical micropollutants, they get entry into the environment via drug manufacturing companies, wastewater treatment plant, through veterinary uses. So through effluent, they come into the environment, they come into the ecosystem. So soil, water get contaminated with this pharmaceutical micropollutant, and at the end they get entry inside the food chain. For this, to avoid the entry of these pharmaceutically active compounds and heavy metals which are leached from the contaminated water into the drinking water reservoirs, we are in the need of in-site remediation. For this, when surface water utilized for groundwater recharge under the effect of sewage effluents, polar drug residues, they might come into the groundwater. So flocculation and sand filtration, it can be used, but they are not that much efficient to remove drug residue. So to avoid this, bank filtration and artificial recharge of groundwater, these are the options. Besides this, bioaugmentation, it can serve as a green technique, which by this technique we could increase the degradative capacity of the autochthonous microorganism as well as augmented microorganism. There are various types of bioaugmentation. It can be microbial bioaugmentation, plant bioaugmentation, and gene bioaugmentation. So abstract of this study: during COVID-19 pandemic situation, acetaminophen and hydroxychloroquine, their usage was extensive. So entry of these drugs into the ecosystem through the soil and water, it causes ecological threat, because these drugs, they are having high water solubility. To avoid the fate of these drugs and their degradative product, it is necessary to find the eco-friendly solution. Bioaugmentation, it can serve as eco-friendly method. So current study was carried out where acetaminophen and hydroxychloroquine degrading microbial consortia was used for the remediation of soil which was artificially polluted with these drugs. HPLC was performed, and based on the HPLC data, it was inferred that Cosaconia covani, Bacillus velegensis, Pseudomonas aeruginosa, Bacillus subtilis, Penibacillus miscellaneosus, they could degrade 89 and 95% of acetaminophen in sterile as well as non-sterile soil. In non-sterile soil, their performance was better as compared to sterile one. Whereas Cosaconia covani, Bacillus velegensis, Pseudomonas aeruginosa, and Penibacillus miscellaneosus, they could degrade 90 and 98% of hydroxychloroquine in sterile as well as non-sterile soil. So here again non-sterile soil showed more efficiency because of presence of native microorganism. In case of hydroxychloroquine polluted control soil, 40 to 50% degradation was observed due to the soil contents. So this is the graphical abstract of the study. For the microorganisms, selected microorganisms were Cosaconia covani, Bacillus velegensis, Pseudomonas aeruginosa, Bacillus subtilis, Penibacillus miscellaneosus. They were used for the bioaugmentation of soil which was spiked with acetaminophen. For hydroxychloroquine, consortia of Cosaconia covani, Bacillus velegensis, Pseudomonas aeruginosa, and Penibacillus miscellaneosus was used. For the preparation, the cultures, they were separately grown in nutrient broth for 48 hours. The enriched broth was centrifuged and biomass was collected. Their cell density was optimized up to 10 raised to 9 cells per ml. Bioaugmentation of the soil was done as per the method of Jajic well and Labu for heavy metal bioaugmentation. So after this preparation of consortia, microcosm was prepared. Uncontaminated soil was collected. It was dried. It was sieved through 2 mesh to ensure uniformity. Sterilized soil and non-sterile soil sets were prepared. Concentration of acetaminophen was optimized up to 1000 milligram per kilogram of soil, and for hydroxychloroquine 10 mg, 100 mg per kilogram soil concentration was optimized. Control sets, they were also maintained. So total 8 sets they were prepared: control, test sample, sterile soil with and without microorganism, and non-sterile soil set with and without microorganism. 10% inocula was added to the soil with using sterile spoons and plates. But soil was checked for bacterial count as well as the concentration of remaining concentration of drugs. After every 2 days interval, SPC was determined. Concentration of acetaminophen and hydroxychloroquine in soil was assessed by using high performance liquid chromatography. For this, methanolic extract of soil was evaporated till dry residue. HPLC was performed as per generic acidic method. Water aligns 2695 HPLC with photodiode array detector was used. Solvent systems used was mobile phase A water acetonitrile fumaric acid in gradient pattern. Gradient elution pattern was used and flow rate was maintained 1 ml per minute. For the data analysis, Empower 2 software was used. Results and discussion. So this is the count of bacterial count in all microcosm set. So as we can see in set E, where control sterile soil is maintained along with hydroxychloroquine, bacterial count is 0. Whereas in sterile soil containing acetaminophen, still there was some microbial count and more bacterial activity or more bacterial count was observed in non-sterile soil spike with acetaminophen and hydroxychloroquine. So this data suggests that microorganisms they follow lag, log and stationary phase. After exhaustion of these drugs, bacterial count was reduced because in these sets, in all these microcosm set, drug was serving as a carbon source. So these are the peaks of HPLC chromatogram of all the set. This is the data of acetaminophen. So as we can see here, in set B and set D, besides acetaminophen, peaks, degradative metabolite peaks were observed. This is the HPLC chromatogram for bioaugmented soil spike with hydroxychloroquine. In set F and set H, where sterile soil along with inoculum, and set H, where non-sterile soil was along with inoculum, hydroxychloroquine concentration was very much declined as compared to the control set. This is the degradation profile of drugs in bioaugmented soil. Compared to acetaminophen, hydroxychloroquine degradation rate was maximum, and this degradation rate was maximum in non-sterile soil, where we can say that native microorganisms they might play important role in degradation along with bioaugmented strains. In control soil, because of natural activity, only 10 to 30% degradation was observed for acetaminophen. But in case of hydroxychloroquine, in control sets also 50 to 60% degradation was observed. Results and discussion. Bioaugmentation of soil artificially polluted with the drugs was carried out to find out the efficacy of the microorganisms to tolerate and degrade the drugs in the environment. Selected strains they found to have the potential to tolerate and degrade 89 and 95% of the acetaminophen drugs after 10 days of incubation in sterile as well as non-sterile soil. Survival of the microorganism was tested with the help of spread plate technique. The results showed short lag phase followed by steep increase in the count. After exhaustion of the drug, microbial count was declined. Significant degradation was observed in the bioaugmented non-sterile soil while compared with the sterile soil. Hydroxychloroquine concentration found to get reduced in control soil also, but more degradation, that is up to 98%, was observed in the bioaugmented soil. The reason behind this, it is attributed to the presence of ferric ions, nitrate content and humic acid content in the environment, which assisted photodegradation of the hydroxychloroquine. So success of bioaugmentation, it depends on abiotic and biotic factors, which includes pH, temperature, type of the soil, concentration of the drug, water and organic matter content, additional carbon and nitrogen sources, size of the inoculum, and interactions between the introduced strains and autochthonous microorganisms and inoculant survival. So conclusion, based on the findings, it can be concluded that the selected isolates have the potential to persist and degrade the drugs in 10 days. For the in-site remediation, current study will be helpful. Formulation of consortia, optimization of abiotic and abiotic factors along with the strain improvement program should be done to avoid the early visible and long-term negative consequences imposed by the persistence of the water-soluble drugs. To study the fate of the inoculants, molecular tools and multiphasic methodology has been recommended. So these are the references and thank you.